So my name's Brio Lehman. I'm an assistant professor at the statistics department at University College London, and it's my great pleasure to welcome you all to this workshop today. The workshop's co-hosted by the Alan Turing Institute, which is the UK's National Institute for Data Science and Artificial Intelligence, as well as the Diverse Data Initiative at Genomics England, which is a new initiative aimed at reducing inequalities within genomic medicine by conducting community-based engagement, building tools that promote equity in genomics, and doing whole genome sequencing for over 20,000 people. And the third host for the workshop today is Data Science for Health Equity, which I'll spend just a couple of minutes talking about before moving on to the workshop proper. So Data Science for Health Equity is a global community of experts and enthusiasts working at the intersection of data science and health inequalities. And our aim is to ensure that the latest research and innovations in data science, machine learning, and AI improve everyone's health, and especially those in underserved populations. We're a cross-sector and cross-discipline community. It's open to anyone with an interest in data science, health equity, ethics, and everything in between. And amongst our members, we can count health and care professionals, academics, policymakers, funders, people working in the private sector, and media as well. So if this sounds like you or something that you might be interested in, then please come and join us. Lots of different ways to get involved. You can subscribe to our newsletter by going to www.datascienceforhealthequity.com. If you scroll all the way to the bottom, there's like a very short form that'll take you 5 seconds to fill in. And in our newsletter, there's all the information that you need to figure out how to get involved with the different activities that we run. You can also follow us on Twitter, where you can see our handle there. And if you want to kind of get involved with like organizing activities or sharing activities that you're involved with, or papers, or so on and so forth, then you can join our Slack workspace, and I will post a link to that shortly. So we run regular activities across a variety of focus areas, which we call our themes. We have 6 themes at the moment. These include health data standards, social determinants of health, infectious diseases, improvement science, and 2 more themes, which are the focus of this workshop. One is the genomics theme, and the second is the statistical methods theme. So this is kind of like a collaboration between these 2 themes. The rationale behind the statistical methods theme, which might not be like super obvious for those who are interested in health equity, is, well, the aim of the theme is to improve our understanding of inequalities and reduce disparities through the development of better statistical and machine learning methods. So I think we're all aware that the factors underlying health inequalities are highly complex and involve numerous social, cultural, economic, environmental, and genetic factors as well. And we're also seeing lots of examples of clinical algorithms being used in practice that actually make existing inequalities worse, typically as a result of being trained on biased data sets. So here are just 2 examples of bias in biomedical research. One of which will be very familiar to people on this call, showing the disproportionate amount of human genetic data that comes from European individuals. So this is from a paper by including a couple of authors that I know will be participating in the workshop today. And the one on the right is a very similar example of human microbiome data. And I'm sure we'll hear more examples of bias in kind of genomic data sets and the downstream implications of that through the course of today's workshop. So the purpose of today, what we're kind of hoping to get out of it, is to explore the main challenges to statistical approaches for improving fairness in genomics and genomic medicine. And our aim is to identify the common barriers to the effective use of statistical and machine learning methods across genomic medicine, and in particular with respect to understanding the effects of inequities in genomic data and in statistical models. So exploring, the impact of the bias that we saw on the previous slide, and how that affects various aspects of genomic medicine. Number two is to detect and quantify these unfair biases and the effects of, that should say, inequity in genomics. So, we're all aware of kind of ancestry biases in genomic data, but other existing biases that we should also be aware of. Number three is to improve the equity of statistical approaches and mitigate some of these unfair biases that we're aware of. And number four is to understand the limitations of statistical approaches in mitigating unfair biases and addressing inequities. So just to dwell on this last point for a moment, inequities in genomic medicine are largely rooted in wider socioeconomic inequalities, and whilst there is an important role for statistics and machine learning to play here, we can't just rely on technical solutions to solve all of society's problems. So we should recognize that there's only so far that data-driven tools can get us, and I hope that we'll have a chance to discuss some of these limitations in today's panels. We've got an all-star lineup of experts from across academia, genomic medicine, and industry as part of 3 panels today. The first being on disease mechanisms, second on disease diagnostics, and the third on risk prediction. So thank you in advance to all the panelists and the chairs for taking part in these panels, and also to the audience for engaging with the workshop. I think it's really important to have a wide range of perspectives at the table when we're talking about these issues. So delighted to see so many people here today. Just some housekeeping for what the workshop is going to look like over the next 4 hours or so. As I mentioned, we've got these 3 panels, the first starting in 5 minutes or so. We might start a little bit early on disease mechanisms. Each panel will last an hour long, and we'll have a 15-minute break between the panels for people to have a quick break, so we don't get too much Zoom fatigue. Just for those who are just joining us now, please note that the workshop is being recorded. In terms of how the panels will run, we'll be using Slido for some audience Q&A, so that the chairs will be asking each of the panels a set of questions, but there'll be time towards the end of each panel to field some audience questions. And so you can use Slido to write your questions, and then you can also upvote existing questions, so we can essentially put the most popular questions to the panel. If you're on Twitter.
Hey, could you put the Slido link in the chat or somewhere?
Yes, thanks. I will do. I will do at the start of each panel. Thank you. And yeah, if you're on Twitter, please, follow Dishy first of all. And if you'd like to do any sort of live tweeting, if that's your cup of tea, then please use the Dishy DSXHE hashtag. So everyone can kind of follow on the conversation on Twitter. That is it in terms of housekeeping. I just see in the chat there's someone asking about the Slido link and password. Just I'll post the link to the Slido just in a moment once I stop sharing my screen. So we're a tad early, but I think let me just check if all the panelists for the first panel are here. I think everyone is here. So yes, I think we'll get going with our first panel for today. And so I'm going to introduce our chair for today before I pass over to for the speaker introductions. So delighted to welcome Professor Carolina Kuchenbecker, who's our chair for this first panel. Carolina is Professor of Genetic Epidemiology at University College London. She's also the scientific lead for the Diverse Data Initiative at Genomics England. Her research focuses on the genetic and environmental risk factors for complex diseases by leveraging the unique characteristics of diverse populations. And she's one of the principal investigators of the PARC-H initiative, which is the largest study of mental illness in Pakistan. Just before I pass over to Carolina, we're working to like a relatively tight schedule, so I'll just ask the speakers to be conscious of this and not to declaim for too long. And please do make use of the raise hand button when you would like to speak to make it easier for Carolina to chair. So without further ado, I will stop sharing my screen and pass over to Carolina.
Thank you so much, Brio. Can you hear me all right? Okay, fantastic. Yeah, it's really a great pleasure to be here, and thank you so much for involving me in this panel. I'm very excited to really present a very, very stellar lineup of people who will be discussing the really important topic of fairness, equity, and mechanisms in genomic research. To start in a very light way, I'll introduce the speakers and maybe hand over briefly to each one of them. It would be fantastic if you could add a little bit more color and also say a bit about what your view, your experience is related to genomics, fairness, and mechanisms. I'll start with Professor Nora Franceschini. She's a professor of epidemiology at the University of North Carolina and a nephrology-trained physician and a cardiovascular disease epidemiologist. Her research focuses on environmental and genetic determinants of hypertension, kidney, and cardiovascular disease in diverse and underrepresented US minority populations who carry high burden of chronic health conditions. Nora, do you mind introducing yourself?
Sure. So yeah, I'm Nora Franceschini at UNC Chapel Hill. So, you know, as I think try to give you a background on why I got into diverse populations. I think this was, for me, was, you know, from the beginning when I was training, there was something that I wanted to do, mostly based on my clinical background and also epidemiology background. You do see there's very large differences across population, the burden of disease. I specifically work on kidney cardiovascular disease, and that's kind of, you know, they have a high impact in particularly in the United States, like, you know, American Indians, Blacks, and some subgroups of Hispanics. So for me, it was no question that that's what I want to do for, you know, almost 20 years ago, and that's what I've been doing. I'm being involved and lucky enough to find good partners in the two very large projects that are genome-wide studies of diverse population. One is the COGEN-BP, that's based on African and African Americans, studies for blood pressure. And the other one is COGEN-Kidney, that we are taking more an approach, more global populations, but also particular, you know, trying to not dive too much into European, but they still had the mixture. So there's a lot of complexities, and I think I like the challenge, and it's being so far very exciting field to be working.
Thank you very much. Our second member of this panel is Alex Chalek. He is a core member of the Institute for Medical Engineering and Science, an associate professor of chemistry, and an extramural member of the Institute for Integrative Cancer Research at MIT. He's also an institute member of the Broad Institute, member of the Reagan Institute, and an assistant in immunology at MGH, and an instructor in health sciences and technology at HMS. And I believe also part of the Human Cell Atlas Equity Working Group. Alex, over to you.
Thank you very much, and thanks so much for having me today. I'm delighted to contribute in any way I can. I guess you gave a rough overview of where I'm coming from. I guess as background, our lab is really interested in understanding the cellular and molecular basis of human disease. And over the past several years, we've partnered with people all around the world to develop and apply cutting edge technologies to gain a deeper understanding of sort of the cellular basis of disease. And through this work, we've become very interested, not just in going places and grabbing samples and doing science, but really thinking about how to enable and empower people to perform their own research, to enable them to take the methods that we've helped to develop to push their science forward. And that's led naturally into some of the work we've done in the Human Cell Atlas, where we've helped to lead some of the equity efforts, which have really focused on equity in action, thinking a little bit less about making sure that there's representative sampling alone, which is something that very often comes up, and thinking a lot more about how we make sure that there's equal participation in research and equal data benefit, which is really challenging to define, really challenging to push for. And so, I won't say that this is my area of expertise. I'm trained as a chemist, physicist, and mathematician. I would just say that I'm somebody who has very much enjoyed taking technologies we've developed, pressure testing them with people around the world, and really listening to those individuals to see what sorts of science they would like to do and how they would like to do it. And that's really informed how we try and approach things. So delighted to join this conversation with the disclaimer that I'm not formally trained in this. I'm just a scientist who's interested in the area and interested in contributing however I can. Thanks for having me.
Fantastic, thank you. My experience is very few people are formally trained in this, but we all end up somehow here. Thanks. So our third panelist is Dr. Amir Kenny. She's a professor of medicine and genetics and the founding director of the Institute for Genomic Health at the Icahn School of Medicine at Mount Sinai. She's a renowned expert in population genetics and translational genomics, and leads a team working on problems at the interface of genetic ancestry, machine learning, and medicine. And over to you.
Thanks very much, and thanks to all the organizers for inviting me onto the panel this morning. Yeah, I mean, you asked for a little color, so I'm trying to think back through the years, and a lot of my interest in the topics that we're going to discuss today, which are how do we really think about aspects of fairness, representation, and sometimes even justice through the entire pipeline of genomic research, from basic research through translational research. Go back to early in my career, and my initial interest was really from a scientific point of view, where early in my career I was working in populations that were really underrepresented in the types of genomics enterprise around the world, and learning things in a sort of a engaged participatory research way that were novel and new in those populations. And so from the early part of my career, it just never made a lot of sense to me that we as a genomics community would spend majority of our resources and efforts and scientific endeavor focusing on a small slice of humans. It really meant that we were likely leaving from a scientific perspective, a lot of discovery on the table that maybe we would never know. And from many other perspectives, not really serving humans in the best way that we possibly could from this type of scientific endeavor. So I'll leave it there for now and look forward to some discussion with panelists as we move forward.
Great, thank you. So last but definitely not least, let me introduce Bogdan Pasaniuk. He is an associate professor of computational medicine, human genetics, and pathology and laboratory medicine at UCLA. He develops statistical and computational methods to understand the genetic basis of diseases, focusing on underrepresented populations, integrative genomics, and biobank studies. And he also serves as a PI for the Center for Mixed Populations and Health Equity, and for the Biomedical Data Science Training Program for Precision Health Equity at UCLA.
Thank you so much for the invitation. It's really an honor to sit and talk to you today. I'm really excited to absorb all of the discussion that we might be having today. I'll keep it very short and just give you a highlight of, or a color highlight, if you will, on kind of my interest in this space. So I'm a computational scientist by training, and I develop methodologies to bring or to understand genetic basis of complex traits. And one of the most fascinating questions that kind of has been on our mind for the past decade or so is how genetics operates or in various different ancestry, various different populations. And now we're on the verge of trying to move genomic space towards translational, and we would want to do it in an equitable fashion. So we're very, very interested in places in which, by looking at diversity, both genetics as well as sociodemographics, can improve our understanding of genetics or genetic underpinning of complex traits, and how can we move that forward in a way where all of our patients benefit mostly out of it. So I bring methods focused expertise, if you will, and I'm really interested to hear the other angles of the story. Thank you.
Thank you very much. I think the breadth of expertise that we are having on this panel really shows and highlights the complexity of studying mechanisms in genomics. I'm glad that this is actually the first topic that we will be discussing, because looking at diversity and representation, I think the consequences, the impact on understanding mechanisms is actually underappreciated and much less discussed than other aspects of genomic research and translational genomics. To begin, I'm wondering if one of you could actually talk us very briefly through the sort of steps of getting to understanding mechanisms in genomics. And it could be, there might be different ways based on the genetic associations discovered somewhere. Unless there's a volunteer, I know who I would throw this one to. Amy, I know you've been involved in both discovery and then follow up. Can you just give us a very high level picture of the sort of steps involved in getting to mechanisms?
Yeah, absolutely. And I'm not entirely sure of the constituency on the call today, so I'll try and start off with this as sort of a big picture, in the big picture. And I'm also a population geneticist, so I'm going to start off from a population genetics standpoint. I think to start, we need to think about human beings on the planet and our own history, going back hundreds of thousands of years to our common ancestors in sub-Saharan Africa, and the subsequent movement and migrations of humans around the planet. The last 10,000 years of human history has been particularly interesting because a number of things have happened, including the agriculture and other types of ways in which we've organized ourselves into bigger and bigger societal structures. The fact that those successes have allowed us as a species to grow exponentially in our numbers. So about 10,000 years ago, there was 7 million humans on the planet, we estimate. Today, we're over 8 billion. And the types of increasing diaspora migration and sometimes forced indentured movement of people around the planet have meant that as a species, although we're pretty young compared to many other species occupying the planet, our human genetic diversity is very informed by a lot of those demographic events. And what it means today is that there are, if we start to look across the human genomics, and particularly if we start to look across a diversity of humans on the planet, we start to see very patterns of genetic diversity that are very informative and really inform the discussion that we're going to have today, which are that most of variants that we observe in any human are common, and we tend to think of common variants as variants that are 1% or above, are shared by almost any human on the planet, although it could be at a higher or lower frequency depending on the specific demographic history of that human. But as you start to go lower down into the site frequency spectrum and you start to consider variants that are rarer and rarer, those variants are more recently arisen and tend to reflect more recent demographic events on the planet and dispersals in humans that live in different places. And that's important for a few reasons. One is because for most diseases, it's not only the genetic component that matters, but it's also the environmental component. We can think about this in many different ways: social determinants of health, built environment, atmospheric environment, pollution. There can be lots of ways we think about the non-genetic factors that impact health outcomes and disease. And the other thing that's quite important, and this really got to the heart of, I remember a lot of discussions about this about 15 years ago when we were really designing very, very large scale genomic studies, is that while we expect the biology of disease in our young species to be very similar across disease and disease mechanisms, there can be some exceptions. There can be processes of selection, for example, at play. But for the large part, most diseases have very similar biological mechanisms. However, the variants underlying the specific genes and pathways can vary and can be at different frequency. And that's what's really driving some of the differences in the genetics of disease that we see across human population groups. In addition to the fact that humans are living and existing in many different environments, so the interaction between the genetics of a particular disease and the local environment also comes into play and can lead to different outcomes. So I'll stop there, but I think that that's one of the reasons why it can be it's so important to think about learning about the genetic and other non-genetic factors contributing to disease in many different populations around the world. And often these linking of variants in a genome to specific health outcomes is the first step in getting to the biology and the mechanisms underlying disease.
Thank you so much. Do any of the other panelists have anything to add?
So maybe I can take it from Emer and move forward a little bit. So on a very basic level, what we're thinking about is whether a particular variant causes disease, a particular genetic variant causes disease in the same way in various different individuals of different ancestries and different socioeconomic backgrounds. So that's one of the fundamental questions, which speaks towards whether interactions between genetic variants or between environments and the genetic variants exist or not for various diseases. The other question that it's at heart of this whole endeavor is whether the variants that cause disease, whether they have the same effect or not in various different individuals, whether they are present at different frequencies or not. As a result, what Emer kind of nicely put together as different types of selections that could occur during the demography. And the third component of it, which is another ongoing open question, would be what in my mind is one of the major discovery of GWAS over the past 20 years of GWAS, in that all complex traits in humans are highly polygenic. So we have to think about it in the context of not just one variant at a time, but in the context of thousands, if not tens of thousands, if not all the way to very complex system like omnigenic model, complex system in which all the genes or all the variants kind of interact or work together to provide risk. So all these questions cannot be answered and there are many more, but I'm just synthesizing 3 of them here, cannot be answered unless we study individuals of various different backgrounds, both on the genetic level or genetic ancestry level, but also on the you know, socioeconomic and social determinants of health. So all these questions will have to be tested in all individuals of diverse ancestries if we want these findings to be propagated to all individuals.
Yeah, I totally agree with this, but I want to add also that some populations that we're studying are actually at the mix, and then can make quite complicated because they come, so you can actually look at specific segments of their genome, and they come from different populations based on whatever references we have. And then in this particular mixed populations, some of the reference we actually don't have. So for example, Native American, American Indian are you know, when we are in the US, for example, there is some subgroups have a very high level of admixture of that. And we actually, if you don't know, even they have reference panels, we don't know this diversity, you know, is going to be quite problematic in terms of applying, you know, this kind of information to, for example, if you want to apply for clinical care. So I was just going to give you a very simple example. For example, in pharmacogenomics, you know, we know that some variants can, for example, be associated with specific side effects of the drugs, you know, and if we're not studying a particular population, could be like, you can say, well, American Indian is a very small population. Well, there is a much larger pool of admixed individuals that potentially carry some of these variants that could be, you know, have a clinical utility.
Thank you all. Alex.
I guess I'll just jump in and add one thing really quickly,
Perfect.
which is to say
yeah,
that
sure, take your time.
I 100% agree on a better understanding of the underlying genetic variants and their distribution across individuals. I just want to bring up one point that was raised around sort of understanding the environmental determinants of health. I think there's a lot that we don't understand, not only in terms of socioeconomic status, but in terms of geography, in terms of cultural traditions, but how all of that influences some of the responses that we see in individuals. And I think that one of the biggest problems, and this is something that we're tackling in the HCA, is that genetics is obviously the substrate on which all cellular behaviors are based, but then you have to really think about where those genetic variants work. So there's a big need to understand cell types and how environmental determinants alter epigenetics and therefore alter the expression of some of those variants to lead to the basis of disease. And I think that one of the biggest issues that we have is that we don't have sort of a unified language for understanding what these factors are, for collecting them, and for using that as the basis for understanding correlates. And I think that one thing that becomes really critical, particularly in a lot of the populations that are already raised, is working with local scientists that are part of the communities that we seek to study to understand what are some of the factors that are going to play a critical role in dictating phenotypes, understanding whether it's local differences in diets or, in maybe it's the use of tobacco. I mean, there are lots of different things that play a critical role. And I just think that being more cognizant of that, not just thinking about the genetics, but thinking about how that might play out in the long run in terms of cellular phenotypes, in terms of exposures, I think that that's going to be really critical to link some of the genetic basis of disease to sort of the tissue level manifestations that go through the cell.
Bogdan, did you want to follow up on both the environment and working with local scientists?
I would like to follow up on obviously all these are absolutely I'm fully in agreement on with everything that has been said. I just want to raise an awareness about one potential challenge that I think we're kind of in this panel. We're supposed to pose all these questions and try to figure things out, solutions for them. When we like to think about this lack of diversity in genomics, we like to think this beautiful work all the way from Alicia Martin and Emer's work, looking at GWAS or looking at the genetics of individuals. But in some way, we have a similar lack of diversity in looking at molecular type of traits. And this goes all the way through gene expression as one molecular phenotype, in this EQTL studies all the way to current single cell studies. So one way to think about it is that type of problem, as lack of diversity in such studies, it's probably a great place to try to intervene and try to prevent the perpetuation of the lack of diversity. The other angle that I would add here, and I'll punt to Emer to hear more about, is I think one next wave of data sets that are hopefully trying to tackle some of the diversity or some of the access to social determinants of health would be biobank type studies, or studies in which all the individuals within a medical system, at least in the US or in Europe, within an entire country, let's say, or all the wonderful wonderful initiatives that we just heard at the beginning of this call, in which individuals through a cross-section of the entire medical system or entire country are linked with all sorts of data, all sorts of diverse medical record data or registry-based data, from which we can learn more about the interaction between genetics, if you will, or genomics and social determinants of health. And I'll raise that as one potential, way that we can try to tackle some of these open questions.
Yeah, so Caroline, if it's okay, maybe I'll pick up on some of the points that Bogdan and Alex just made to try and help us think about what's going to happen in the next 10 years in the field of genomics. So one of the points that Bogdan just made is what's really happening a lot around the world in this shift of the generation of genomic data. I think in the last 20 years, we have seen most of genomic data is generated in sort of academic, researchy environments, sometimes also in industry. But I think one thing that's really going to be a big sea change in the next 5 and 10 years is we're going to see a big shift to the majority of genomic data being generated by health systems. And many of those health systems have research entities like biobanks, which are linked to electronic health records and other medical data, which can be used for the secondary purpose of research. Even though that's not necessarily how that data was collected, it is a very powerful translational research engine. And it's not true that biobanks represent more diversity than the kinds of GWAS catalog-y databases we have out there yet, but they do have this feature that they do tend to be more population-based. Anyone who walks into the health system, less designed, less selected, and that health system and resources around the world are starting to generate these entities. We wrote a review on this a couple of years ago, and most of the biobanks were really sitting in North America, Europe, and certain parts of Europe actually, and certain parts of Asia. But we are starting to see many other health systems and biobanks coming up in many other areas around the world, and many researchers and physicians behind those great efforts. So just by that sort of acceleration and change in or evolution maybe in the way that genomic data is being generated, and the link to a fuller medical phenome through health systems data, and the way health systems data in general are digitizing their data, are linking their data to other outside data sources with an understanding that health systems have a bigger role in population health management. Those things are really, I think, going to have a big impact on the type of data that gets generated, the accessibility of that data for researchers. I think we have to be also a little bit creative about thinking not just in centralized way, but also in federated ways. The other point that I think was Alex and Bogdan mentioned was molecular data. We're still, I think, in the not at a population scale way of studying molecular data. And I'm not dissing great and wonderful projects that are out there like GTEx or the Human Cell Atlas or many others, but they're really looking at data at a cellular level and cellular phenotype, or sometimes tissue level data, and the order of hundreds or even thousands of individuals. There's a big gap between that and what we can do with genomic data, which gets up to hundreds of millions of people. However, quickly coming down the pipeline for bringing this types of observations to population scale. Methylation, I think, is going to be one of the technologies that we bring at that intermediate phenotype at a cellular level that captures the environmental component in addition to biological components. That is really going to start to scale up pretty soon. And we'll start to get some insights. I mean, what we learned through genetics is that in the early days of genetics, when we're thinking about association, I'm not really thinking about linkage or family study design now for a moment. I'm just thinking about association. Is we really needed to get to study samples, you know, in the millions almost, to get rid of some of the signal to noise and to start to see that full polygenic signal of disease. And I think that we're going to need numbers certainly much larger than we have today to start to get rid of some of the signal to noise in the molecular phenotypes and get more robust understanding. Obviously, molecular phenotypes are more complex because you can look at any cell in the body, and we're increasingly getting resolution on cell types, but it's still complex from that point of view. And then just last point, and then I'll shut up and leave it to other people. But I think Alex, I think it was mentioned this, really thinking about the importance of engaging with local researchers and importantly, diversifying the STEM workforce. That's essential. I think, you know, our scientific STEM workforce, at least those of us who work on genetics, looks very much like the databases we have generated for genomics research. And that's part of the limit, because we lack the knowledge or the prioritization or the insight sometimes to ask the very important questions that impact other populations around the world. So generating resources, training pipelines, and support for others to develop their own resources is going to be a very important part of this endeavor. And I'll stop there.
Thanks, great. I think it's really helpful to think about the different types of data that are necessary to get to mechanisms from just, you know, discovery of some forms of association. I know, Nora, you've been also doing some work looking at diversity and epigenetic data resources. Feel free to comment on other parts as well, okay. But it would be great if you could talk a little bit about your work in that area.
Sure, yeah. So I first want to make a comment, and I think Mark came with a lot of important points, but I want to put a plug also for cohort studies. So biobanks and electronic, you know, hospital-based or healthcare-based are excellent for example, to collect a large number of diseases that are probably not as common. But there is issues into the data quality in terms of like things that Alex talked about, social determinants of health and things like that, more environmental factors that we are interested in including in our research. So there is issues on that. So there is also huge opportunities in continue doing cohort studies that are more population-based. Healthcare is a selected group of people. They go take care of that particular facility, and there may be some of them are, excluding maybe the population that have, the poor population that not be able to afford care. So just putting a plug for cohorts here. But going to your question, yeah, we did. We have it's fantastic work actually from Charles Breeze, currently NCI, and he looked in the ENCODE data, and the question was a very simple question, just quite similar we've been doing GWAS, is the ENCODE data, so in cells and tissues epigenetic elements. Is there samples that we have in that repository that all of us use for annotation? We also use for fine mapping, and I know that we probably have some methods that incorporate some of these elements, regulatory elements in the statistical analysis. So when we look at that, and is mirror just what we have in GWAS and EWAS and etc., there's just a very small sample, diverse population. I have to say that I'm an optimistic person. So actually, when we did this work, we look and I identified there's like 60 Hispanics samples with high quality and up-to-date technology for, connecting open and things like that. There's also some African samples generated recently. So I was just, my thought was we just need to expand this, but we also need to use the resources already there.
Great, thank you. Alex, you're obviously working really in this area of, the molecular phenotypes and so on. One of the things that would be great if you could comment on, does it really matter if those come from ancestrally diverse samples, or, given as Amir said, the molecular mechanisms are likely, fundamentally the same. So do we care whether we have diversity in cell lines, experiments, epigenetic resources, etc.?
So I do not think that they are fundamentally the same. I would challenge that. I would also, emphasize this point that I think the molecular phenotyping, the kind of stuff we do is great, but I also don't think it scales in the way that was brought up before. I think that what we really need to do is to think about how to interdigitate different types of data sets in order to generate and test hypotheses. If you can do things like, genomic sequencing on the scale of millions, tens of millions, hundreds of millions to find variants of interest, then the question becomes, which of those are accessible, playing through the epigenetics, understanding how that plays out in different populations. And you might say, why would I think that it plays out differently in different populations? It's because we have data that shows that it does. You get access to different inflammatory stimuli. Imagine you're a cell in the gut, you have a different microbiome, you have, exposure to different insults, you have different amounts of cytokines and chemokines acting on you, which leads to a different epigenetic state, which causes molecular pathways to play out differently because you have similar transcription factors at different levels, but maybe in different complexes and also binding to different areas. And so at the end of the day, that leads to different phenotypes that become very clear. I think that one of the issues is that our cells are so plastic, they're made to adapt to environments. And so it's not surprising that they're going to try and find a homeostatic set point that will be different in different contexts, and that over time, you'll see a far enough drift that you could say these really aren't similar anymore. And I mean, we've seen things that, that are very different in different contexts. Like we've seen stomach cells show up in the intestine, in some studies that we've done with partners in Zambia, just given environmental insults. And so I don't think it plays out similarly. And I think understanding disease mechanisms requires that kind of resolution. But I also think that you can't do this on everybody. You have to get the variants that you want. You have to understand how environmental stimuli are going to alter the underlying genetics and how that's going to play out as cellular phenotype to get to the piece. So I think it's sort of going from the genetics at the large scale to systematically working your way through how it might actually be driving a disease phenotype or risk phenotype. But I also think that it becomes critical in that, once again, to connect back to the community to understand what are the types of insults that might be relevant for environmental exposures. And I'm also pretty optimistic. We've done a lot of stuff in the past where we've tried to play with environmental exposures to modulate models in the lab. And so I think once we understand what it is we're trying to phenocopy, there might be many ways where we could go about doing that and begin to understand what variants would do. Our ability to read and write the genome really changes our ability to ask directed questions of how specific genetic variants lead to changes in function at different levels. But in order to do that, there is some degree of profiling and there is some degree of bringing together these pieces of information across different levels. And so I think that one of the biggest concerns that I have is that, there's a different language and different strategies being used across different disciplines. And so very often the people that are focused on genetics and genetic fairness aren't necessarily talking to the people that are thinking about things at a cellular level, aren't necessarily talking to people that are thinking about epidemiological studies. And I think that in order to do these things right, we have to create a common language and the basis for understanding and sort of unifying those insights. And once again, I'd say to me, a lot of this goes back to also engaging with communities. And the other thing I would just highlight here is I think we're getting a lot of information, and I think it's great to think about, how we can be fair and how we can make sure that things are accessible and we can, try and understand variants. But I do think that, we have to constantly bring it back to this question of, well, if the goal is to understand how differences lead to differences in manifestation of disease, how do we make sure benefits are accruing to the individuals that are disproportionately affected by this? Because very often these things end up benefiting the people that they should not be benefiting, in my opinion. I'll stop there before I keep on speaking.
Bogdan?
I wanted to change a little bit the direction, but I would not jump into the trying to answer the question whether the variants are broadly the same or they have broadly similar effect or not. It's a very fascinating question. And I think it speaks a little bit towards the phenotypes themselves. Cellular phenotypes are in many ways, different than the phenotypes that you observe complex in humans. And I think it's a fantastic question to dive into. The other comment that I wanted to raise to the table for all of us to think about, and I'm biased a little bit because I'm thinking more in terms of the statistical methodology space. I would say that we think a lot about, equity. We think about diversifying data sets, and we think a lot about including or outreaching to all the individuals of all different trainings and different parts of the world, which is terrific. All these things need to move forward. I also think that we need to start diversifying the methodologies themselves. It's often the case that the statistical methodologies that we use have been developed and fine-tuned with a single population in mind or a single genetic ancestry in mind. They've been optimized, greatly for Europeans, let's say, or European ancestry individuals, mostly Northern European ancestry individuals, and then they are just ported to different ancestries or different contexts. And it's often the case that they are not working as well, simply because they've been optimized for some context and not the other ones. There's an entire literature, there's an entire area coming mostly from machine learning domain about fairness in statistical methodologies from the beginning of the conceptualization of the mathematical model all the way to implementation. And I think there's a huge area from statistical methodologies perspective to incorporate all these concepts of fairness, of equity, of making sure that the methods have equal or robust performance across all different contexts from the beginning stages and not just an afterthought after the methodologies have been developed. So this is an area that I'm a big fan of it, and hopefully hopefully there are more that want to join that part of it.
Oh, it's really helpful. You are helping us focusing now a little bit more on the statistical methods side of things, right? Because data and input is one thing, but yeah, methods is another. Do you or anybody else have specific examples of methods or analyses that are currently, that are tricky to apply to a more genetically diverse population or different types of data sets? So where are some of the key problems and what do we have to change?
I'm just going to give a very small plug because I'm not a statistician, but I'm the user of what's generated. So I think any method that require reference population and require that we have the exact LD is very problematic when we're dealing with admixed populations. So that would be just my small comment on this.
I would just give the most boring but fundamental one in all of this, which is to say that anytime you want to look for a feature of statistical significance, you need a background distribution. And insofar as your background distribution is biased and you don't explicitly model that, it's a problem. And if you don't have enough data, so you end up bootstrapping, that's also a problem. So I think in each of these things, we have to make sure that we have a sufficient amount of background across different populations to understand when a variant is actually, associative, or when a feature is of relevance. And I don't think that we build enough of that in. And so once again, I think it comes down to doing appropriate metadata annotations and understanding factors that will play a role there. And obviously, if you're doing it at the genetics level, it's different than the epigenetics level, it's different than the transcriptional or proteomic level, but just making sure that there is sufficient background to test hypotheses. Because otherwise many things will come up as significant that are not significant, or some things will be underweighted because you end up having too much of one population as your background population.
Yeah, maybe I can continue a little bit. I'll just also give like a positive kind of encouragement towards that. So probably one of the best example where looking at diverse genetic ancestries towards being able to more accurately pinpoint causal variation or putative causal variation, I should say, just based on statistical, is trans ancestry fine mapping, where differences in the linkage disequilibrium and variant frequency patterns can be used to pinpoint or to remove out tagging a variation versus the variation that is more likely to explain the association signal. And based on all sorts of different metrics, you could argue that that's one of the most powerful signal, the differences in trans ancestry linkage disequilibrium, to pinpoint these causal variants. I would also say that on the topic of admixture, I think this is a little bit of an underappreciated topic, and I'm really happy that, Nora, you're raising this. Some numbers that kind of come into our mind, especially coming from LA, is that within the US alone, roughly 1 in 2 people are going to label themselves as having mixed genetic ancestries within the next decade or so. And that's all true worldwide, that genetic ancestries get more and more complex. And the idea of an admixture population being a simple model of 2 ancestries, and then a fixed number of generations, and then the genomes being a mosaic of segments from 2 continental sources, it's probably just a first step and a very rough approximation. Admixture happens on a continuous scale, happens between all different communities, all different, and presumably all of us that live in larger cities see that every day. And in my mind, that's one of the biggest questions that needs to be solved, because there we see diversity at the genetics level and the non-genetic level within every individual. So every individual have its own unique patterns that needs to be disentangled. And I'll stop there in the interest of time.
Yeah, I'll add. I think this this idea of admixture that both Nora and Bogdan have brought up is a very important one, but it's also a very fundamental one, because you could make an argument that every human on the planet is admixed for some choice of source populations, and we tend to see the impact of admixture when source populations tend to be more genetically divergent. So, for example, African Americans in the US, or admixed African populations in Africa. But it's true of everyone to a certain degree, and we see this as we actually go deeper into the site frequency spectrum. So it's the type of complexity in the genomic landscape in populations that we encounter. The other point I'd like to raise is, I think maybe some of this got a little bit missed in some of the conversation, but I'd just like to reiterate, I don't think anybody here on this panel is saying that we expect the variants underlying disease to be the same across populations. We know that variant associations are going to be different across populations. What we're saying at an aggregate level, at a disease level, we don't expect, for the most part, the biology to be wildly different, but the variant associations, whether they're at a cellular level or at an organismal level, can be very different. And that's the landscape we're in, and that's the challenge of biology and genetics. But it is interesting to think about how we integrate this data. And, again, just thinking about what has happened, particularly in the last, I would say, 10 years, what will likely happen in the next 10 or 20 years, is that we will have a combined landscape of very large data sets that maybe are more full in terms of the medical phenome or links to environment at an individual level, but not designed for any hypothesis-based question. At the same time, we have studies that are really focused on specific hypotheses that are controlled and designed to be able to answer that question. And to me, what gets very interesting, and in ways in which we can think about how we can redress some of the ways I think we went a little bit wrong or off course in the last 10 years is to think about how we bring those pieces of information together. I think when we try and think about designing study cohorts for epidemiology, we're really trying to design them to ask specific questions, but there's ways in which the insights from those cohorts can be made more generalized, and the idea and questions of transportability and generalizability and fuller clinical context can start to be addressed by linking that information with other more population-based cohorts. And that brings me to my last point, which is something that Bogdan mentioned, the example of trans ancestry fine mapping kind of gets down fundamentally to the question you're trying to answer this comes a lot from the field of machine learning. If you're trying to understand more robustly which variants are driving the association, or you're putting into your genomic predictor, then bringing in more diverse populations with LD that starts to break down some of the confounding in that observation is really the right thing to do. If you're trying to be more empowered to link variants with disease, then maybe building out more single population resources, the thing that we should do. And that was the thinking behind a lot of what we have been doing in the field of genomics. I would argue that maybe we should be thinking about other metrics in addition to accuracy, sensitivity, specificity. Also, we should be thinking about robustness, transferability, generalizability, portability when we're really discussing biological findings.
A couple of questions from the audience that link directly to the methods. One is, can you give specific examples on where population differences may play a role, or is it mostly about linkage to equilibrium? And the other one is, should those methods incorporate ancestry on a continuum? Are we going in this direction, and what are the challenges there?
Bogdan, you might like to take this one.
Yeah, I could take a stab at it. This is this is an open question, so it's definitely up for debate and up for folks to. I don't think the questions have been fully answered, but I'm of the personal belief that most of it's linkage to disequilibrium and frequency. Most of it is the vast majority of lack of portability of polygenic risk scores, or lack of portability of genetic findings from one population to the other one, has only have to do with the variant themselves having different frequencies in different contexts, and or not being at the right causal one, but just at the tagging one. And there's some works out there, and we have some works as well, where we can try to quantify that. And I'm of the belief that the vast majority of lack of portability is explained by just that, and not by the effect size varying, the causal biological effect per allele varying across ancestry. The other angle with the continuum, yes, I think that's really one of the most fascinating. And again, I'm biased because we actually work in this space in my group. I think we need to start moving away from clumping individuals into genetic ancestry groupings that are often very arbitrary. They're very, I urge everyone to start reading this high impact papers when they group individuals across ancestries and try to understand why there's a circle drawn somewhere in the principal component map and not a little bit further. So I do think that on the statistical methodology space, we need to start thinking about every individual being in a unique position in a genetic continuum map. This is something that was raised in the past couple of years by many works from, I think, folks in this audience. And I do think that statistically we can actually do that for some applications. That being said, when we think about predicting, genetic risk, we do want to incorporate population back into it, because at the level of population, we have measures that we would need for converting genetic risk into an absolute or relative scale, something like prevalence, something like other factors that might impact that population. So there's room for both. But I do think there's a need for more smarter statistical methodologies that look at a continuum and not at, very homogeneous blobs of individuals. I'll stop there.
Yeah, I just want to make a comment that we did that in the whole genome sequencing in the top med contest, and you actually put all the individuals together and account for as much as you can. And I have to say that the gain is in terms of significance is much smaller than when you subset groups. So it tells me that you're correct. We do need to change some of the methods. We're doing that and not stop analyzing all them together, but instead just have better methods to do that. I am going to put myself in the spot, and this is a method that I love, is colocalization. But I have serious problems with people using colocalization in the mixed population or across populations, and you do see this very often. I think colocalization, one of the assumptions that you actually know the LD between the whatever you're colocalizing. And this is being one of the methods that I always have to think to before I apply. I would actually love to know the opinion of other people on that.
Great, thank you. I just wanted in the last minutes maybe look a little bit towards the future. My impression is that in genome-wide association studies, ancestry or the lack of diversity, it's increasingly appreciated, reported at least, and more often also criticized. But it seems to be more of an emerging issue in all of the other related data sets and methods surrounding it, especially the complex work that helps us uncover mechanisms that usually doesn't just involve one group of experts, but people from different fields, more molecular statistics, working on cell lines, working with model organisms. And the ancestral diversity, for example, in the data sets and resources that are being used there is often a lot more hidden. It's not necessarily reported. What's the one thing you feel we should be focusing on addressing in the next 5 years or so? If you had a lot of money or a lot of power, even more than you already have, what do you think would be the one thing that moves us forward, could move us forward more than other things?
I mean, I can start and I'll try and keep it brief. I've been very encouraged by efforts from big either federally government funded entities, or sometimes these efforts happen more locally on the ground. And I'll shout out maybe H3 Africa is a really good example of this, to develop resources and training and sort of and data and expertise, and really sort of focus that on the ground in areas of the world where this type of infrastructure is new, and in building that infrastructure, really creating an environment where that really focuses really local talent to start to develop their own scientific communities, insights and findings from local communities, informed by local communities. And, really the type of speed at which such efforts result in the acceleration of deep scientific environments and the bringing together of local research entities. There's many other projects that are doing this. For example, in South America, there's a number of groups who are standing up their own long read sequencing hubs, technologies, resources in order to develop foundational resources for the field of genomics in those areas of the world. And some of what's helped this come along, as well as being a broad movement towards generating policy guidelines and federated resources within the genomics community that can be shared to anyone else in the world. And I'll shout out again, J4GH is a really good alliance that has been doing this for the last decade and more. More of that, however, needs to come. So it's very encouraging to see what has been enabled, but with comparatively not a huge amount of investment, I would say. But I think that more of that really needs to come. And if in the next 10 years, we could see a lot more investment into generating resources in a way that's disseminated to many areas of the world, that to me would be would have all sorts of anticipated and even unanticipated benefits for the genomic community.
Great, and just given the time, if the others could also maybe give us their take on the sort of vision for the future, what we need to address, but possibly stick to a minute or two. Sorry about that. That would be great. Who would go next?
Maybe I can be super fast. I'm fully agreeing with Emer. I think that's one of the area that would be the most fruitful in the next decade. The genomic data set will get diversified by the nature of things. We're already on our way there. I think building the infrastructure that goes beyond the few countries where this is done, genomics as well, towards worldwide and really syncing with the local communities, it's probably the best use of any type of resources.
Yeah, I'll go the next. I think, I agree with the prior speakers, but I think at this point, we do need to invest in junior investigators. We need to fund them. We need to support them, and mostly the creative that want to, address all these challenges that we have ahead. That this will be a fantastic use of funding. And I think Alex maybe is the one that brought that. I think we need to actually engage also the broader community, how important this work is, and what we think is going to be the benefit in terms of for communities and for the population in terms of either medical or even in knowledge, in terms of knowledge. Or mechanisms, for example. So I think the investments in junior investigators for me will be, the trigger for all these other things that we want to do, and obviously generate more data so they can use that.
So really quickly, I'll just echo what everybody else had to say, particularly the engagement of scientists around the world, both in terms of their training, but also engagement of local communities to make sure that some of the benefits trickle back. I think that, additional emphasis on training and on, partnering to collect references, but thinking not just about sort of FAIR principles, but CARE principles, really thinking about giving people control over their own data, thinking about responsibility, thinking about those elements. I think we have to incorporate that. And I think, one of the biggest things that I think about is how we can fold some of this into the training of the next generation of scientists, not just around the world, but even in our own labs and locally. I often say the biggest impact that we can have in science is how we teach people to do science, as opposed to anything that we would do ourselves. And so I would emphasize that thinking about how we could get training to be thoughtful about these matters into standard curricula so that people are thinking about it ahead of time, so that when they're building models, it is natural for them to ask questions like this, would be one of the things that I would really emphasize. I think it's great that we're talking about this in a panel, but I think that if it was something that was intuitive to people to ask how some of these confounders might play a role in some of the analyses that they're doing, that would be phenomenal and fantastic, because it would really help to move the field forward.
Thank you. Before I thank all the speakers, I just wanted to point out that there are lots of great questions in the chat, and I didn't actually manage to bring in a huge number. If the panelists are still around for a bit, it would be fantastic if you could maybe just answer them on the chat. And then I thought it was an absolutely fascinating and insightful discussion. A huge thank you to all speakers. I particularly liked how at the end, we really came back to some of the root causes of inequity, and with a very broad picture, and covered even aspects of training and who's doing the research. So thank you all very, very much for this fascinating discussion.
Thinking of how we take this area of genomics further forward, and so it's an absolute delight to have our three panelists here today, who I'll introduce very shortly. And as with the previous session, it'd be fantastic if you could add a little bit more to my introduction to yourselves, and also to kind of expand that into your interests and your experiences of developing genomic approaches for disease diagnostics. We've got an incredibly broad panel, which I think is fantastic, all of whom I'm sure, as we'll find out, are hit by the same issues and have kind of common goals to solve over the next few years. So first of all, I'd like to introduce Professor Alison Curran, who's a professor of medicine and epidemiology and population health at Stanford University. She's the Associate Chief of the Division of Oncology, the Director of the Stanford Women's Clinical Cancer Genetics Program, and co-leader of the Stanford Cancer Institute Population Sciences Program. So Alison, I'm going to pass over to you just to kind of expand a little bit on that introduction, and also to give us some insights into your experiences to date.
Wonderful. Well, thank you so much for inviting me and for that kind introduction. As Dr. Ellingford said, I am at Stanford and have been for the last 2 decades. And I'm also a clinically active oncologist, where I treat patients with breast cancer, in addition to seeing patients in our cancer genetics program, together with genetic counselors for diagnostic testing to determine inheritance of pathogenic variants in various genes. My research focuses on genetic epidemiology and outcomes of primarily breast and gynecologic cancers, although my recent work is encompassing germline genetics and genetic testing of all cancer types. And through this experience, I've become very interested in understanding whether the remarkable advances in genetic knowledge and technology have in fact translated into improved cancer prevention and treatment at the population level, and also in identifying gaps in this translation so that we can target them for intervention. My colleagues and I have identified racial, ethnic, and socioeconomic inequities in genetic testing and in the clarity of genetic testing results in the United States. And these experiences have really shaped my interest in using data to improve fairness in genomics and precision medicine. Thank you.
It's fantastic to have you here. Finally, it's a delight to introduce Professor Nancy Cox, who is the Mary Phillips Edmonds Gray Professor and Director of the Vanderbilt Genetics Institute and the Division of Genetic Medicine. She runs a computational lab focused on developing and applying methods for understanding the genetic components of human disease, and also works with a multi-institutional group, including the Vanderbilt University Medical Center, the Meharry Medical College, and the University of Miami, to develop innovative solutions to chronic disease health inequalities. Nancy, I'm going to pass over to you, if I can, just to expand on that introduction a little bit, and to also give us some insights into your experiences and your interests in applying tackling these sorts of questions in disease diagnostics. I think you're on mute.
The big thing that we're interested in right now is tackling what I think of as institutionalized health disparities that relate to the use of everyday laboratory values, an astonishing proportion of which had their reference ranges set decades ago in what would now be considered small samples of white men. That has played out over the years to lead to non-European populations bearing a disproportionate burden of repeat testing, but also over and under diagnosis of disease, over and under use of medications and procedures. And we used the Vanderbilt electronic health records going back an average of 10 to 15 years in 2.8 million patients, but up to 30 years in some individuals, as well as our biobank of 300,000 samples of DNA, to really look at the consequences and potential solutions to this problem. And there's really no way to solve this problem without using genetics. This is not about race. Race is a social construct, and you can't tell from a person's color what alleles they have with respect to some of these actually quite large effects in many cases. More than half of everyday laboratory values, so about 70%, are heritable, and a third of those are highly heritable. And physicians think of these laboratory measures as purely dynamic measures of disease risk and progression. And when you're looking at some of these highly heritable measures, the genetics is affecting the staging. It's definitely true that there's a dynamic component to these laboratory values, but the genetics can hugely affect the staging, and accounts oftentimes for people who progress rapidly to disease from health, or conversely, never progress to disease despite being staged as if they have an illness. And I think the fact that it is non-European populations that bear the disproportionate burden of this means we've got to tackle this problem. There is no way to do this without bringing genetics into it. And the truth is that the number of laboratory values where the genetics is predictive of the outcomes that we're looking for is actually in the minority. Most of the biomarkers we use would be better biomarkers if we regressed out the genetics and used a purely dynamic measure for the disease progression studies. There's no one size fits all solution to this problem, but none of the solutions really can be instituted without the genetic information on people undergoing the laboratory testing.
Absolutely. So that leads quite nicely to kind of my first direct question to you as panelists, which is, what are the main disparities and inequalities that we see in disease diagnostics? And where do these arise, and how do they arise as part of the diagnostic pipelines that we apply? So if I pass that one to you, Alison, first, and then we can go around the other speakers as well.
Absolutely. So I will tell a bit of an example or a bit of a story from my experience, both as a doctor and as a scientist in the area of cancer genetics. And so what we see in the US, and I'm sure Dr. Cox is familiar with this, is that there have been disparities in access to germline genetic testing. So for example, for BRCA1 and BRCA2 gene mutations, and now a slew of other genes for which we are routinely testing. And that's largely due to the exorbitant cost of these tests, at least in past decades. It is getting better, although not as much as we'd like. But so because of this access disparity, over time, there has become an information disparity. And so what we see is that people of ancestries other than white Northern European are far more likely to receive an uncertain result on sequencing. So when there are 3 results that are given clinically of pathogenic variant in a gene like BRCA1 or BRCA2, negative or normal, and then the third category of variant of uncertain significance, basically that this hasn't been well codified and we don't really know whether or not it affects the protein. That is far more common among people of ethnicities other than white Northern European, because similar to what Dr. Cox was saying, of whose genetic sequence was originally defined as normal. So it's quite interesting. It really is this access disparity perpetuating an information disparity. And on some level, that wouldn't matter if everybody would just ignore these uncertain results and not act upon them. But we and others have found evidence that sometimes they're in fact leading to inappropriate care, even preventive surgeries with removal of organs. So that obviously is a big problem. And I think it gets to be more of a problem as we sequence more and more genes clinically, and genes that aren't as well defined and referenced across populations. So we certainly don't want to stand in the way of progress, but we do want to ensure that this disparity doesn't grow based on these historical inequities. So that's the vignette that we're seeing in my field. And I think there's much discussion and concern about how to address it.
Fantastic. And that raises some really interesting points that I think we'll come back to later around how we could best apply these massively parallel assays to understand variant effect, and particularly those that have been applied to these types of genes like BRCA1, where we still see disparity. And so finally, Nancy, if I come to you for that first initial question, so what are the main disparities and inequalities that you see from your experience, and where do they arise in the diagnostic pipeline?
So a common challenge is the most commonly used blood test, which is the full CBC. White cell counts, especially and neutrophil counts in African Americans, are usually lower, in part because of the Duffy polymorphism, a well-known, totally benign polymorphism that leads to lower circulating levels of white cells, but not lower total numbers. So there's no greater degree of infection to these low white cell counts. On the contrary, this is perfectly normal, and it's an adaptation to a particular form of malarial parasite in Africa, where that allele is nearly fixed in the African populations where that parasite is common. So in African Americans, with an average of 80% African ancestries, 20% European ancestries, about 2/3 of African Americans are homozygous for this polymorphism and have low white cell counts as a consequence. Those people are kept out of cancer clinical trials because they rarely meet the standard for the blood count. Nobody's checking Duffy to see whether that's the reason they don't meet the standard for the blood count. They are taken off chemotherapeutic agents that are cytotoxic much before they need to be, and therefore are not receiving the full line of therapy they need. That's true also for a number of autoimmune conditions where first-line therapies like azathioprine are used. We've always known that African American women do more poorly with lupus, for example, than European women. It turns out they get to take about 20% of on average, of first-line medications, as European women do, before they're taken off those meds, almost always just because of a low white cell count, which is both unnecessary and counterproductive to the long-term outcomes. And it's not, so it's, they also get, African Americans in the US also get, when the only reason for a bone marrow biopsy is low white cell count, the biopsy is never positive in those who are homozygotes for the Duffy polymorphism. So there's unnecessary bone marrow biopsies being done in the US a lot for what is a benign polymorphism. African Americans also are more likely to carry a G6PD, an allele for G6PD deficiency. Carriers, this is an X-linked trait, male carriers have on average 1 point lower of hemoglobin A1C levels. And in our biobank, that is misleading patients and their physicians into believing their diabetic control is better than it is. It probably leads to a longer time until the diagnosis is made. That leads to a 1.2-fold increased risk of any diabetic complication, but particularly 1.8-fold increased risk of diabetic retinopathies and 1.4-fold increased risk of diabetic kidney disease, just in people who are carriers of G6PD deficiency. And that's because there's a red cell hemolysis that happens, so the red cell turnover is faster. So it's not measuring hemoglobin over the usual 3 months, as in Europeans. It's measuring the glycosylated hemoglobin over a much shorter period of time. And that's why the apparent glycosylation is less. So these are really common ways in which US minority populations are getting less good care. And as I say, if we use the genetic information, we could fix this in a way that could create much greater health equity, reduce numbers of unnecessary tests, and improve the healthcare system, reducing the number of unnecessary bone marrow biopsies, increasing the availability of minority populations to participate in in cancer clinical trials. And finally, to be able to get the full benefits of the medications we use. I mentioned cancer, and we have the data for lupus, but transplant does not look good either, for exactly the same reasons. So I think these are such good wins for so little investment. It's crazy that we persist in largely ignoring this problem.
And such impactful wins that would have such an impact on these people's lives as well.
Yeah.
Okay, so that leads us actually really nicely onto the next section, where it'd be great to talk a little bit around kind of intersectionality. So this concept that it's not just one or two groups or categories that you could put people in that drives that disparity in access to testing and access to equitable testing, but a combination of these different factors. And so I guess the question that I think is worth discussing is whether we're using the right, and meaning this in the most sensitive way that we can, the right categories to group individuals in when we're being concerned about genomic diagnostic tests. And so in your experience, are we using the right categories, or should we be expanding to use other information? And from that previous answer, you certainly talked about using genetics as an opportunity to categorize individuals and potentially enable targeted or tailored precision medicine programs. But are there any other things that are worth considering in that context also? So if I pass that one to you first, Nancy.
So I think it is critical that we do a better job in medicine of capturing social determinants of health. It's been difficult in medical centers to put those into play in the US because of the need to protect patient privacy. And so all of the geographic measures of things that contribute to health disparities, like degree of particulate matter, our EPA does great measurements of this, and those can be linked to geographic coordinates quite well. But for patient privacy reasons, we often can't instantiate those directly in the electronic health records, except by not the exact location, but a much broader region. Similarly, for social determinants of health that we know are important, that relate to easy access to healthcare, the food deserts that can be a prominent factor in levels of obesity, for example, which is a major risk factor for disease. Right now, research studies have often been able to instantiate these, but we really need to have them in hospital records and as part of routine medical care. Another example is early childhood trauma, which is a tremendous risk factor for long-term bad health outcomes and neuropsychiatric disease, but separately. I mean, all kinds of long-term health outcomes are affected by early childhood trauma. And with climate change, introducing all kinds of weather and climate-related traumas in coastal regions, we really have to get this under control and be collecting the necessary information to be able to use this appropriately in healthcare. So that's definitely one thing that we need to do better. But I also think the access to having genetics is a little bit crazy. And certainly the United States is a melting pot. And I'm deadly serious when I say you can't look at a person and know whether or not you should order a Duffy test on them, right? We have so many people who are actually turn out to be Duffy homozygotes who classify themselves as white and in fact have substantial European ancestries, but they have enough African ancestries on both sides that they're Duffy homozygotes, and they have low white cell counts in exactly the same way and for the same reasons with the same consequences. And so we have to stop thinking of this as being about what people think of as race. It's about alleles that we have. And if we use the genetics, we can do this just fine without having to go to these crazy classifications that don't really have a biological basis, when what we're trying to do is biology and medicine.
Mm-hmm. And so, Alison, if we come to you next to reflect on your experiences there. So the direct question here is, are we using the right categorizations in the context of genomic diagnostic tests? And perhaps we could elaborate that a little into when it's appropriate to use self-reported versus genetic ancestry.
Well, I think it's a very important question. And just to reflect a little bit on the excellent comments of Dr. Cox there, I'm sort of thinking about the situation of oncology. And just to, again, give an example of what I think happens, we do see this problem that was mentioned of lower white blood cell count, ethnic neutropenia, as it's sometimes been called. That's sort of a silly name, but lower white blood cell count occasionally being an issue in terms of oncologists being afraid to dose appropriately for chemotherapy. And there's intersectionality there with the problem of obesity. So because of obesity being more common in certain populations, again, due to food deserts and other problems with access, we also see oncologists dosing low for body mass index, being worried about dosing at the right levels for weight because the number seems too high. And so I think we get this problem of a few different factors, some genetics, some about social determinants of health that play into worse care for some people. And then, of course, when you bring in genetic diagnostics, such as BRCA or other genetic testing, revealing uncertain results that might prompt inappropriate use of surgery and other interventions, we see a complex picture, I think, much of which really has more to do with structural racism and other problems in America related to social determinants of health than actually genetics. But some of the genetic factors, I think, play into it in a complex way. So I think in terms of thinking of things in terms of racial categorization, it's less that than sort of teasing out the different factors that come together to create suboptimal care, and how do we pull out each strand and work on trying to improve it.
If I come to you next to discuss the role of non-genetic factors and how we can handle inequalities of these factors.
So I mentioned social determinants of health, which matter a great deal. And of course, the better we understand that, the better position we're in to use societal means to reduce the kinds of exposures that increase risk of disease. Really, part of the value of genetics should be helping to throw non-genetic factors into stronger light. Almost everything that we look at is a G by E. And even in the rare space, it's really striking that structural birth defects have a much lower solve rate on average than other rare N of 1 sort of kinds of diseases, which really suggests the importance of exposures, a G by E, driving some of those. And we know that there are drugs that are teratogens. That's a very common source of structural birth defects, as exposure to teratogens during pregnancy. But there's almost certainly much more of that than we appreciate, in part because of things like the maternal genotype interacting with the fetal genotype in the context of an exposure. So if you imagine a slow metabolizer mother and a slow metabolizer fetus, there could be profound consequences of a drug exposure in that combination that is normally perfectly well tolerated. And so this is what I mean by an opportunity to use genetics to learn more about the exposures that matter, so that we can help to reduce the likelihood of those exposures, particularly in the people most vulnerable. I do think the United States and other countries, the few of them that depend on private insurers for so much of healthcare, are in a more challenging position at getting real equity in terms of access to genetic and having the ability to get genetic information. And And in part, so Hillary mentioned, as we build out better quality models so that even private insurers can be assured that we're capturing sort of everybody and that we have basically the same solve rates, a very predictable solve rate for everybody that we capture, with models built to identify everybody who really does need genetic testing. That helps to even the playing field so that insurers are more confident that their investments will actually pay off in the long run. But it's very hard in the US, where the average person is 5 years with their insurance company before they move on. You can see that it's harder for insurers to justify the kinds of investments that pay long-term benefits like genetics. And that's just something we have to find a way to deal with in our society.
And Alison, if we pass that same question to you.
Yes, I certainly couldn't agree more that we have many challenges in the United States with our healthcare system, and it's a frustration to many of us every day, and I certainly wish it would change. I think, again, while that is a problem that we all struggle with here, there are interesting, almost natural experiments of sort of what happens when you improve access, and I think they can be learned from. So again, I'll give an example from my field, which would be looking at BRCA1 and 2 genetic testing in the United States. So that was protected by patent for a long time, about 20 years, maybe a little bit less. In 2013, we had a United States Supreme Court decision that ended that. And very quickly, prices fell dramatically. So as I recall, it was about $3,500 in the morning and, $1,500, by the next day almost. I mean, not quite that dramatic, but nearly. And subsequently, we've seen panels of genes fall in cost to somewhere around $200 out of pocket. Now, that's still quite a lot for some people. I'm not saying it's good enough, but there has been a dramatic fall as the market basically opened. And with that, we have also seen accumulating knowledge about the prevalence of some of these gene mutations, BRCA1 and 2 in particular, but others as well. And we've seen greater coverage by insurance based on broader guidelines for who should be tested. Now, again, this is challenging as we bring in newer and newer genes, or not newer genes, but more newly tested genes that are poorly understood, and we get more variants of uncertain significance there. But when you look at the genes that are best understood, so for example, BRCA1 and BRCA2, we've actually seen the uncertainty rate, the disparity in uncertainty between racial ethnic groups go down. And so we've done some work on that, showing that over time, again, we are seeing a lower rate of variant of uncertain significance disparity between racial and ethnic groups. So I think in a way that's sort of a natural experiment, right? That when you do improve access and you start to be able to get a bigger data set, maybe not necessarily for the right reasons, but just the market opening up, you do see a loss of this gradient to at least some extent. And so I think we need to do better with that, but it's interesting to see that as access improves, we do see some of the improvement in terms of the uncertainty in results.
I think we have to work together as a community to try and tackle all of those challenges. Okay, and Nancy, to come to you for a final comment in the minute or so we have left. The biggest frustration that I could hear from you was the ability to perform genetic testing and get access to those results when ensuring recruitment into these different trials. So what do you think is the major step that's inhibiting that, and what sort of impact do you think, and I know you gave us some clinical utility stories, but what sort of impact do you think that would have at a population level?
So I think one of the things that this is about is that in introducing genetics has increased health inequities inadvertently because not everybody has the same access. But I think the data from the laboratory values exposes that we already have institutionalized health disparities that we need to undo. Deciding whether or not to do genetic testing for rare conditions and cancer is one thing. That's new science, new growth, new kinds of health technologies. We have institutionalized health disparities in medicine now that we must undo. That is And to do that, we need genetic testing in everybody that can be used in basically creating more equitable lab values to use in everyday medicine. And I was qualitatively different. This is using genetics to reduce existing, long-standing health disparities. And I think once you know this, you can't unknow it. And I think we have to do it because we are doing harm. First, do no harm. We are doing harm in medicine every day because we haven't undone this. So it's very hard. It's really hard to work on anything else until we get this fixed, honestly.
I think that's a really, really powerful closing remark, actually, to summarize a lot of the things that we've talked about. So thank you very much to all three of you. That was some immensely insightful, important discussions across a variety of different areas, but also different environments that we all live in. And so I'm going to pass back to Dr. Lehman, unless there's anything else to say, but just to thank you all once again for those really interesting discussions.
Thank you so much, Jamie. That was a really excellent discussion, and thank you to the panelists. Also, there were a few unanswered questions in the Slido. So if the panelists do have a moment to have a look and maybe pop some answers in the chat, if that's straightforward, then that would be great. So we've now got just under 15 minutes until our final panel for the workshop. So I'm going to just pause the recording. Forget. So it's my great pleasure to welcome our chair for this final panel, and that's Dr. Sam Lambert. Sam is Assistant Professor of Health Data Science at the Department of Public Health and Primary Care at the University of Cambridge. Sam has research interests in polygenic scores and co-leads the development of the polygenic score catalogue, which many of us here will be familiar with colleagues at the EBI, where he's also a visiting researcher. So I'm going to pass over to Sam to introduce the speakers for today. Over to you, Sam.
Yeah. Thanks, Priya. Yeah, so we'll get started with this panel on risk prediction and equity and risk prediction. We'll go through the speakers first, same format as last time, where each speaker, I'll give a little introduction and they'll tell us a little bit about the research before moving on to questions that I've prepared. And then please do add to the Slido while we're going, and we'll take up your questions at the end. So I don't know, yeah, we'll start with our first speaker. Are they here? Is Rahib here? I can't see his video. So our first speaker is Rahib Ali, who's the Chief Medical Officer of Our Future Health and a consultant in acute medicine at the Oxford University Hospitals Trust. They're also a clinical epidemiologist at the University of Cambridge and director of the Public Health Research Center and associate professor at New York University, Abu Dhabi. And so maybe Rahib, if you could give us a little introduction to your research and your work.
Sure, thanks, Sam. So I'll keep it brief so we can have plenty of time for further questions and discussions. But I think the reason I've been invited today is mainly based on my role as Chief Medical Officer of Our Future Health. And for those of you who are not familiar with Our Future Health, it's a fairly new research program that started in the UK last year. And the aim of Our Future Health is really to understand how best to detect common chronic diseases early, intervene early, and thereby prevent disease. And one of the key parts of that early detection is the use of genetic and genomic information, particularly polygenic risk scores. So Our Future Health aims to recruit 5 million people amongst all adults in the UK, so 18 plus, from all different ethnic groups and from all socioeconomic backgrounds, because one of the key limitations of previous studies, not just in the UK, but globally, has been underrepresentation of those groups. And so we are aiming for a very large sample size of 5 million people, which is about 10 times bigger than anything that's been attempted before in the UK. But we've made a good start, and I'm happy to talk more about what we're going to be doing with our future health after the other speakers have introduced themselves as well.
Great, thank you for that introduction. And it was great to see you in the Times on Saturday and your full spread. That was very cool to see science really taking a center stage. Yeah, so our next speaker and panelist is Gil McBean, who's the founder and chief scientific officer of Genomics PLC. He has a background in statistical and population genomics and played leading roles in international HapMath and the 1000 Genomes projects, as well as advisory roles in projects including the UK Biobank and the Genomics England company. He was founding director of the Oxford University's Big Data Institute and is a fellow of both the Royal Society and the Academy of Medical Sciences. So Gil, why don't you introduce the research that you're going to talk about?
Thanks, Sam, and thanks to the other organisers of this workshop for inviting me. It's great to be here. So I am one of the founders and the Chief Scientific Officer of Genomics PLC. We're a UK-based company, about 120 people with offices in Oxford, Cambridge, and a small outpost in the US. So we're about realising the information that's carried in our genomes to improve health in its broadest sense, and kind of one of the primary ways in which we do that is to understand through things like polygenic risk scores the risk that we all carry for a broad range of common and complex diseases in our genomes, and particularly in the context of the other types of information that we can measure about ourselves, to understand what that means in terms of future risk for a broad range of disease, but also potential benefit of possible interventions. And so in that context, it's clearly critical to think about issues to do with fairness and equity. We're absolutely driven by the idea of giving benefit to everyone, but also, as you can imagine, given not just the state of research and the way in which it's been focused over the last decade or so, particularly with respect to genetics, but also actually the broader field of medical or epidemiological research and how that has traditionally not delivered as much insight to underrepresented populations as, say, people who look like me. It's clear that we are in the business of addressing issues to do with how fair are we and how can we act to reduce inequality. And so, although I'm sure we'll get into this a lot in the detail, when we as a company are developing our insights, our products, our services, which are all based on essentially trying to bring together a planet's worth of genomic data, we have to be really careful about measuring the performance of these products and services by issues to do with context. Of course, ultimately, that's what we're looking at here, is how do statements that you can make about risk and benefit translate into statements that are both accurate and powerful for people in different contexts. And so I'm sure these are many of the themes that we'll explore in this setting. You know, we're sort of committed to measuring. I think that's one of the key things. We're committed to validating and improving our technologies across a broad range of contexts, whether that's age, sex, ethnicity, ancestry, geography. But we also recognize that there are gaps and there are holes in where we are today, which we as a broader community and us specifically as a company have to work to address. So I really look forward to the rest of this panel discussion. I think it'd be really interesting to hear a broad range of views on this topic. Thanks, Sam.
Great, thank you. Our next panelist is Segun Fatimo, who's an associate professor of genetic epidemiology and bioinformatics at the MRC and UVRI Uganda and London School of Hygiene and Tropical Medicine unit in the UK. He leads the African Computational Genomics Research Group at the MRC UVRI and at the London School's unit in Uganda. So Segun, if you could give us an introduction to your research.
Okay, thank you very much, Sam. And thank you very much for inviting me to be on this panel for this workshop. So what I do mainly is to try to use genetic data to find some association or causation between our genetic information and different kind of disease. And I primarily focus on a range of cardiometabolic traits with special interest in African population. What we have realized in the last few years is that African population is underrepresented in genomic study. And thanks to so many initiatives, in the last 10 years, focus has been primarily on how to improve representation of African in genomic study. You may be aware about the H3 African initiative. So what that has done is to improve that representation to some level. So since I became independent as a research person about 3 years ago, my focus also has been on improving representation. So currently, I'm leading the Uganda Genome Resource, which has been, when we published that paper about 4 years ago in 2019, represent the largest genome study in African population. And that was just about 7,000 individuals compared to what you will see, for example, in the UK Biobank and other population. So currently, we are now also generating what we call the Malawi Genome Resource, which is very, very similar to the Uganda Genome Resource in terms of design, also in terms of phenotype that are available. So Malawi is in South Africa. And I'm also co-leading a program that we call the Nigeria 100,000 Genome Resource. So this resource is being developed also, first of all, to improve representation and to be able to do all the interesting things that I want to do, including polygenic risk score that we mentioned. So basically, it's mainly basic science. And we know that at some point we'll be able to move from basic science to see whether there can be clinical utility of polygenic risk score in African population. But first, we need to improve representation. Thank you.
Thanks, Segun. Our next panelist is Nilanjan Chatterjee, who's the Bloomberg Distinguished Professor of Biostatistics, Oncology, and Genetic Epidemiology at Johns Hopkins University. And prior to joining Johns Hopkins, he was the chair of the biostatistics branch of the Division of Cancer Epidemiology and Genetics at the National Cancer Institute in the US. His research focuses on the development and application of novel methods for the analysis of large-scale genetic and genomic studies, including those from diverse populations. So Nilanjan, if you want to introduce your research in the area. Oh, you're muted.
Yeah, I'll keep it brief so that we can get started with the exciting panel. I do methodological research in multiple areas of statistical genetics and beyond. And my interest in polygenic risk scores goes quite a bit back, starting from around 2008 or 9, when the genomic studies started emerging. And one of the things I was interested in earlier was, like, in the beginning, there's only few genetic variants, and then when people are evaluating the genetic scores, we saw that there's probably only modest utility. And I got interested in trying to see that how, as we do larger and larger studies, how do we expect this performance of genetic risk score to improve, and connecting it back to this polygenic architecture of diseases. And we provided some, I think, comprehensive picture of how performance of genetic risk score is going to change over time with sample size and also as the disease architectures. And then we are also interested in developing methods, to improve genetic scores using different types of information, including functional annotation data or various types of machine learning techniques. And the other area I was always very interested in, like how these genetic scores will be used for clinical utility. And there we always emphasize the importance of you developing absolute risk models, which can not only incorporate the genetic risk score, but can provide the context in the presence of environmental risk factors and other non-genetic factors, biomarkers, but also taking into account, the heterogeneity in disease incidence rate, outcome rates across diverse population, which I believe is going to be extremely critical. And when you are talking about equity, we just cannot talk about equity unless we talk about the context, and the context is really different by the different diverse populations. And then more recently, we are also been developing algorithms for how to improve the basic performance of genetic scores into diverse populations using data from multi-ancestry genome association studies. And we are also working on that area.
Great, thank you. So yeah, let's we'll pick up the conversation, or maybe the context area is something to think about first, because I think when people are talking about context, they might be talking about different things. In terms of context, you might be talking in the UK versus the US, but context also might refer to maybe subgroups of individuals, maybe by ancestry, ethnicity, and things of that nature. And I don't know, does anyone want to pick up on that? What do they think? What are they talking about when they mean context? And do you mean always just these categorical differences, or are there other contexts that you think are sort of more continuous in nature?
I suggest you call us out.
Okay, well, you've spoke, so you have to speak first. I know Lingen raised his hand though, so he's safe to go.
Yeah, I think both kind of context is important, and I think like we are realizing more and more that sort of self-reported categorization of populations based on self-reported ancestry or other information has no biological basis. And if you think in terms of genetics, we are probably better off defining, not categorizing people, and into you start using more continuous definition of genetic ancestry, which has been shown to be the variations we have in terms of genetic risk is actually more continuous and not discrete by the categorical groups. But at the same time, we have existing inequity in the system, and whether if you look at, for example, disease incidence rate, or incidence rate for various diseases, or the survival after disease. And these are definitely very variable by self-reported definition of ancestry, which is not to say these are biological definitions, and they are biologically meaningful, but whatever component of the health inequity is there, for example, what happens survival after cancer incidence, right? That is highly determined by healthcare access, economic cost, and that has a basic structural inequity. And so I think like when you're trying to show the performance of polygenic risk score in the bigger context of this heterogeneity, we will need to keep using the self-reported definition, because those things does correlate with the various factors like disease incidence rate, survival, and the healthcare cost, and so on. And so we need to, I think, be defining metrics like evaluate the performance of these genetic scores combined with other factors, thinking about outcomes which will take into account all of these different factors. Otherwise, for example, if we just model everything in terms of disease risk, then we might lose the picture that there's unequal consequence of the same disease diagnosis across diverse populations, so in terms of risk and benefit of these interventions. So that's what I think that that we will probably continue to need to use both of them into our modeling framework.
Rahib, I saw you wanted to respond or add on to that.
Yeah, no, sure. I mean, I think the point about what drives differences in disease outcomes by different ethnic groups, it's historically been obviously quite difficult to answer because when you compare countries, there are so many differences in the way that diseases are registered, access, etc. The UK has a big advantage in that access in general is not different across different ethnic groups or different socioeconomic groups in the main. We do rely on self-assigned ethnicity because up until recently there were no alternatives actually So it's not as if we had genetic ancestry to look at for most of these analyses, but some of the work that I did previously was showing how incidence of different cancers varied by self-assigned ethnicity within the UK. And there were very significant differences, up to 5-fold differences for certain cancers, which were not explained by known modifiable risk factors. So these are the kind of research questions which we would be unable to answer unless these new studies, UK Biobank, for example, which I also worked on, many years ago as a junior researcher, we had tried to oversample different ethnic groups in order to be able to look at some of the outcomes, but that wasn't possible at that time. Now with our Future Health, because the sample size is so much bigger, and if we can get 1 million people of non-European ancestry, would have sufficient statistical power to look at those outcomes, not just for cancers, which actually are much rarer, of course, but for more common diseases like diabetes, ischemic heart disease, etc., where, again, we know there are differences primarily by self-assigned ethnicity up to now, but we will be able to look at the impact of genetic ancestry on those in our Future Health. So I just wanted to add that point.
Yeah, I think that's really interesting. And then is there targeted recruitment, or is it by virtue of sample size that you think you'll achieve sort of representativeness for the UK? Because I guess and do you have quality indicators? Because I feel like that might be quite helpful.
The UK is probably the only place in the world where you could attempt to do this, because one, we have a very multi-ethnic population, and the last census showed that 18% of the population is of non-British white ancestry. Although about 5% are European ancestry, but it's got a very large non-European ancestry as well. And the second thing is because there's a single system through the NHS, and we can link to all relevant or main data sources, whether primary care, secondary care, cancer, death, etc. We'll be able to link those outcomes to both the phenotypic and genotypic data that we have, where everyone in the country, the study is open to everybody in the country, as you read on Saturday. But we do recognize that in order to get representativeness, you do need to make extra efforts to recruit people, not just from ethnic minorities, but particularly from more socioeconomically deprived backgrounds and some geographical areas within the UK where historically they've been underrepresented.
Gil, you had your hand raised.
Thanks. In the context of risk prediction, and particularly risk prediction using genetics, I think it's helpful to try and be really clear about what it is that we want to achieve or want to avoid, conversely, and why context, including race, ancestry, ethnicity, and so on, is important. And I think it's helpful to make a distinction between two things. One is something we want to avoid, which is bias. It's really critical in the context of risk prediction tools that there is not systematic over or under prediction of risk, or conversely benefit in the context of therapeutics, on the basis of aspects which are clearly aspects of an individual which should not be linked to access to healthcare or anything like that. So there's a really important issue around avoiding bias, and certainly some of the earlier work around transferability of polygenic risk scores really noted how there were issues, very systematic issues of bias, and with respect to genetic ancestry. I think the field's got a lot better in sort of addressing that issue, but nonetheless, addressing that issue with respect to the genetic ancestry. But there are also other areas of context. Age is one I'm particularly interested in. Socioeconomic status, I think, is another very important one, where there are aspects, there is differential performance, shall we say. And what's not totally clear to me is whether that then translates to bias. So the other aspect that we should care about is the sort of the performance. How good are you at identifying people who will benefit or not from a particular intervention? And then I think there are some really important questions about what it is that we should be trying to achieve. We all want to maximize outcome, and we probably want to minimize inequity in outcome. But we also need to acknowledge that different groups come, has already been said, with very different baselines for risk. So for example, if you take type 2 diabetes, the polygenic risk score in Europeans is incredibly powerful at sort of discriminating between high and low risk individuals. If you look at the sort of odds ratio per standard deviation, if you like, for the PRS in people of South Asian ancestry, genetic ancestry, it's less good. It's not as discriminatory for all the reasons that we understand. But actually, because the baseline rates in South Asian populations for type 2 diabetes are so much higher, in terms of the number of people you can identify who would be above some absolute threshold for risk, the sort of the combined integrated risk prediction is actually more powerful in South Asian populations. Now, I fully acknowledge that doesn't translate necessarily to equity of outcome, because you then have to do something with that information, and that whole downstream piece is perhaps the piece that we're not focusing on enough.
Yeah, definitely. Yes. Shagun, did you have anything to add, or do you? Because I think you've thought a lot about whether different models, different polygenic scores work equitably in different sort of ancestries within, or different fine-scaled ancestries within sort of the larger African population.
Yeah, that's just to follow up on what Gil was saying. In one of the studies that we did on genetic risk score of type 2 diabetes in African population, of lipid traits and type 2 diabetes in African population. So what you will even find is that when PRS is predicting into African population, so it's not predicting the same way between two different countries within Africa. So you see one predicting better, one predicting very, very, very poorly. And what that means is that so there cannot be one standard fit all for PRS for different population in Africa, considering the level of genetic diversity even within African population. And now I'm not talking about between African population and European population, but between two African population. And that also, we have seen that again and again in one study that we are currently doing. So you would expect that the genetic architecture between Kenya and Uganda will share the same boundary is very, very similar. But we see something that is a bit very surprising. We're still trying to explore that. So what I mean to say is that there cannot be that standard genetic risk score that can fit into different population in Africa. So if I could make a comment on different assets. So when you were talking about access to health and others, so one thing that came to mind was if you think about a disease like a sickle cell disease, for example, which is very, very, very common in African population. About 90% of prevalence of sickle cell globally is within just 3 countries. That's Nigeria, Republic of Congo, and India. But when you look at the treatment for sickle cell disease, maybe sickness such as maybe bone marrow or gene therapy, you don't find them in that population. You find them somewhere else. You find them in the West, in the US, and in the UK. So you see, the access, as in my opinion, should be where it is most needed, not where the money is, and that is very, very key.
Since it's a statistical session, I wondered if anyone wanted to comment on whether, on Shagun's point about whether there might not be a single polygenic score genetic risk model for, like, whether it might have to be population specific, or whether whether improved methodology or improved data sets might change that, so there's one model for everyone, or whether those two can't be reconciled. I don't know if Gil or Nilanjan have thoughts on this subject.
Happy to voice an opinion. So my view on that is, let's start in a different place. I think the empirical evidence, as Shagun and others have said, is that with current methodologies, there is frequently a phenomenon of lack of transferability. Now, what does that point to? There are a number of different factors at play. One is the fact that there are different genetic architectures for the same trait in different places. That is clearly the case with type 2 diabetes is an excellent example. Different genetic risk factors contribute differently in different places. And I mean, you see it in some extreme cases where there are rare mutations that cause extreme versions of a common disease, which are strongly enriched in particular isolated populations. And so there are definitely issues of genetic architecture. The second point is that there is a lot of genetic drift between populations, and because we have imperfect fine mapping, and we have to rely on things like imputation, we have some, we don't necessarily have exactly the same phenotyping, for example, in all populations. That can lead to what you might call technical differences in performance between populations. That it's not because the biology is different, it's just that your underlying measurement technologies don't work equally well in the different places, and therefore you don't, the information doesn't transfer particularly well. But then I guess my third point is the fundamental sort of belief, and I think it's backed up by a lot of evidence, that actually there is despite those two things, there is an awful lot of shared biology. Humans in different places are broadly pretty similar. There's a lot of common variation that affects traits whose origin predates the separation of major populations within human populations, if we want to talk about populations. So what that means to me is that you need to recognize the importance of the locality and the population in which you're working, but also you should be bringing together the benefit of having a planet's worth of data to bear on that one individual sitting in one specific place. And I think there are some good methodologies out there, and in genomics we've also been working on this problem, which allow you to say, okay, here's a planet's worth of information, but it's measured in different contexts. Here's the individual in a particular context. How do you weight, for want of a better word, the different bits of information to maximize what I can say about one individual? And what we find is different traits, you need slightly different ways of doing that weighting because of differences in these underlying things, like the influence of the underlying architecture through to differences in phenotyping, through to imputation, and so on. So I don't think there's a simple answer, but I do think fundamentally we want to be using all of the information possible to bear on any individual, but recognizing differences.
Nilanjan,
Sure. Yeah, I'll add a couple of more points on top of that. One is like, I think the data availability issues, because the way a lot of the summary statistics data are available, they're already kind of categorized into ancestries, and that sort of, then we are kind of, we are boxed into developing methods where that will try to use, this kind of categorical categorized data, GWAS data, and then a lot of the current methods are focused on how to take all of the data and borrow information across the different populations to develop maybe an optimized genetic score for each types of populations. And it's an interest now. So that might be the reason that a lot of the methods are going towards that, because that's the way the data is available. But if we have more and more individual level data where we can take advantage of more global and local genetic ancestry information, the methods can take into account more continuous genetic ancestry information to come up with a more optimal PRS for each person which would be optimized for that person, keeping taking the context of that person. But an interesting question is that whether we'll be able to develop a one robust PRS versus you have multiple PRS for like one for each distinct population. Like if one of the goal is kind of developing a robust PRS that will perform well across different population, then we have to think about what is we are trying to optimize, right? I mean, our standard statistical method, we do not have a fairness constraint in the algorithm that we try to optimize that let's say maximize the R square for this particular population, that particular population. But if you're trying to develop an overall PRS, and then we may have a R square for the overall population, but then we may add a fairness constraint so that you say like, okay, you have to also more equalized performance across populations. And I haven't seen that kind of use of. I mean, there is a machine learning literature on that. There is quite a bit of work on that in the machine learning field. I haven't seen the use of those kind of techniques for PRS methodology, but I think at least thinking of a framework of kind of bringing this fairness constraint directly into the genetic risk score development could be useful, I think, for coming up with robust PRS that will be working across diverse populations.
And also, and like you said, sort of not using sort of binary labels, but allowing like maybe ancestry as a continuum as a sort of a constraint there as well.
Right.
So it sounds like the methodology does.
The data availability, right? The data have mostly been available in the summary statistics format. So then we have limited ability to use that continuous genetic insensitive information. So it can be hopefully in the future, that we'll have more actual individual level data for developing these models.
Mm-hmm. Yeah. Yeah, so it sounds like there's a lot of fruit in terms of or way to go in terms of making methods more equitable across ancestries. I guess since we should acknowledge that this is a mantle of all men, we should also say that there's a lot of inequities also in other aspects like sex and gender, and these I think might come into cause with genetic risk prediction as well, where prevalences are quite different between men and women, and also in some cases, diseases will have maybe a sex-specific genetic architecture, and I wonder if people have thought about that, or if they have any thoughts on how sex and gender or other non-genetic or social constructs might play into how we construct polygenic scores, or the methodologies we use to evaluate them in.
I think some of the same principle applies because we know, for example, diabetes incidence rate or heart disease incidence rates are very different by gender. So even if let's say genetic risk score has the same odds ratio per standard deviation unit, but just because the incident rates are different, that will mean different proportion of population will be identified at the same risk threshold. So that's why when evaluating the clinical utility of this genetic score by different groups, whether gender or other social groups, it'll be really important to keep those context into the modeling. And I do feel like the thinking about absolute risk framework and integrating that with information on genetic risk score, but all the other relevant factors, including gender, race, self-reported race, and other social determinants of health is super critical to think about how whether in a particular context, how will genetic risk score will be useful. And also, I think it's also important to show like the incremental utility of genetic score on top of, for example, age or, BMI is a very strong predictor for type 2 diabetes, and few risk factors, sometimes very strong risk factor, allows strong level of risk stratification. And these are easy to measure, so it will be also important to show that like what is the incremental utility of genetic score in those contexts.
Rabe, did you want to add next?
And
I think your clinical.
Well, I was going to focus really on the socioeconomic aspect of this. So again, for diseases like type 2 diabetes and ischemic heart disease, as I just mentioned, the polygenic risk score is a relatively small component of how we would calculate an individual patient's risk, particularly at older ages. And what we will be providing back to participants in general would not just be their polygenic risk score, it would be their integrated risk score, taking into account their age, BMI, blood pressure, cholesterol, waist circumference, etc. So those integrated risk scores, again, are based on data that's being collected often from somewhat biased populations or non-representative populations, particularly by deprivation. Those groups have always been somewhat underrepresented. And so although they may not directly impact the polygenic component, they would impact the overall integrated risk score. And so again, within our future health, we are trying to ensure that we get adequate representativeness from more deprived groups who, as we know, have the highest rates of obesity, diabetes, heart disease, many cancers, and much lower life expectancy overall. So it's in many ways, it's the biggest determinant of outcomes compared to all of these other factors.
Gil?
Thanks. There's another aspect to this, which is to do with need, I think, or the potential for genetics to make a difference. First of all, I fully agree with everyone that integrating the context in which genetics resides is a really critical piece of their deployment, whether it's age, sex, ancestry, ethnicity, socioeconomic status, etc. But there's a sort of important question about whether one should be driven by where there is genetic information that is powerful versus where there is actually a need. So if you, to take a somewhat trivial, well, not trivial, but a real example, the PRS for Alzheimer's disease is astonishingly powerful. You really can discriminate between people very likely to get the disease or not. But no one's particularly talking about rolling that out at a sort of clinical level because you don't know what to do with the information. I'm not saying there's nothing you can do, but it's clearly not very much today that one can do. It's certainly in the way of prophylactic drug treatment. So where genetics is powerful is where it can reveal hidden risk that we can actually act on. We can actually act on today. And I think the community broadly has tended to be slightly more, oh, where is their genetics? How strong is that PRS for this trait? Rather than a sort of need-driven, like, where do we see inequalities, or where do we see the potential for better risk stratification within clinical practice to actually make a difference? And then find/generate the genetic resources to make that happen. And maybe there are some quite sort of weird and wonderful corners of medicine that could be totally transformed by having a really strong PRS that we kind of haven't thought about because there aren't big GWAS communities who've been doing that piece of work. And whether it's sort of osteoporosis or VTE or something, both of which, I'm not saying there aren't people working on GWAS of those, they both clearly are, but that's not the sort of cardiometabolic space where there's been a lot of activity. So I think there's an important call for the community to actually be a little bit more need driven rather than, oh, we've got this amazing light of genomics, let's shine it and assume that that's going to transform healthcare. So it sort of plays into this sort of issue of how should we think about bias or differential context. One of the sort of missing pieces, I think, at least for me, is okay, where are the big gaps, and where should the community really be focusing its effort to try and address the problems?
Thanks. Yeah, I think that really echoes, I think, what Nancy Cox was saying in the last session, sort of about using genetics to sort of contextualize or like sort of make clinical medicine better and like making current measurements that are unfair according to ancestry or genetics better. In terms, yeah, she was using the sort of example of blood counts, but I think, yeah, focusing on where we can sort of use or target genetics to make risk stratification even better if that's an undeserved need. And I think it's sort of what Shagun was saying about how we do know some places where inequalities exist and we're not targeting the resources there as well. right. I think, did anyone else have anything to add on that one?
Can I make a quick comment about the distinction between risk stratification and risk discrimination? And I think we have seen for a number of common diseases like diabetes or heart disease, the current polygenic risk score can, at least for European ancestry population, can provide significant risk stratification in that sense that we can find small fractions of the population who are pretty high risk compared to the average risk population. But that is not same thing as having a very strong risk discrimination in the sense, for example, your AUC value to be very high, like 80% or 90%. So in that sense, we should keep that in mind that just because we have a high risk stratification, that does mean that we can identify small fractions of the population who are pretty high risk, and they could be guided for certain intervention. But not having a high risk discrimination, it also means that a lot of the disease in the population is going to happen outside that small high risk populations. So it's not going to be transformative for the whole population. It's going to benefit a small fraction of the population, which is good because these people can actually do something. But at the population level, still many disease will happen outside these high risk groups. And so the other side of the story has to be told also, that this polygenic risk score has limitations, and in that they do not have very high discriminatory performance. They do have for some common diseases, they have good stratification power at this point.
I wonder, Rahib, if you put on your clinical hat, do you think there's a sort of like a gap there in terms of communicating these complicated sort of measurements in terms of absolute relative risk, and is there more methodology that needs to develop there so that we can sort of evaluate scores better in order to communicate them better to patients and public?
There certainly is, in my experience of talking to fellow clinicians. Many clinicians don't understand these concepts well either. It's not an important part of medical school. It's a relatively small module in most medical curricula. And many doctors do struggle to understand basic statistical concepts because of that. So I think the first step in many ways, it depends who's giving the message. If the message isn't being delivered, whether in primary care or secondary care, it's essential to train doctors first to understand these concepts, and other healthcare professionals to understand these concepts before they can communicate them. But there is a second level, of course, which is the direct to participant, which a study like Our Future Health will be doing at at large scale, at least in the UK for the first time. And we are spending a lot of time and effort now to think about how best to communicate this information back to our participants, where amongst the general public, there is relatively, as you'd expect, relatively limited understanding of these types of concepts, whether it's the role of genomics in disease as a concept in itself, or as you said, relative risk and absolute risk, and how best to do that. We're looking at, of course, I'm not going to name any companies, but companies that are doing this already. Other studies, which I will name, like All of Us, are doing this in the US. But again, in the study like All of Us, they've invested a huge amount of money and resources to provide individual genetic counseling to participants, and that's not easily scalable, and it's not really cost effective, and it's unlikely to be necessary for some diseases. So for polygenic risk score or an integrated risk score for diabetes and cardiovascular disease, I don't think that will be necessary. Whereas, of course, we're moving into diseases where genetic counseling would be required, or when we're looking at highly penetrant rare variants, for example, where genetic counseling would be required, and there's a familial component, then that would involve at this stage having genetic counselors. So for Our Future Health, we're going to start with diseases like diabetes, ischemic heart disease, where that's not necessary, but spend a lot of time developing the appropriate online materials that people can use. And in the second phase, we will be looking to implement something more similar to All of Us have done for some of the other diseases that we'll be looking at.
And I think there's an audience question, particularly for you, about I guess a certain portion of your risk is attributable to genetics, and then a certain proportion could be measured using other means, socioeconomics, lifestyle factors, wearables, diet, things like that. Do you think there's a need like are you only thinking about providing estimates or risk back as a single estimate, or how much, or do you tell people how much is due to genetics versus these other things we can measure at the current stage? Or do you think these are both useful concepts, or should they only be reported in sort of integrated situations?
So in the first instance, as I said, for a disease like diabetes or ischemic heart disease, people are, not all people, but people over the age of 40, for example, in the UK, there is the NHS health check where people do get some of this information back. So they're told this is your 10-year risk of diabetes or your 10-year risk of ischemic heart disease. And to add in the polygenic component to that would be relatively straightforward. Gil and his team at Genetics PLC have started to do this in ischemic heart disease to show how we can feed back those integrated risk scores to patients in the northeast of England. I think it's, in terms of what participants can expect to receive from our future health and similar studies, I don't think it's unreasonable that they would have access to the genetic component because it's their data. But in terms of clinicians, I think we would be using those integrated risk scores in the main, because the risk of disease that's relevant from a clinical point of view, of course, is the combined risk from all of these components, not just the genetic part. I don't know if Gil wants to comment on that.
I can make a comment on that. I think the clinical utility of PRS, I don't think it's going to be across all complex disease. There will be some disease that you can see the utility of PRS, and there will be other that may not be utility. And even where there is utility, I guess what will be very common is that you see maybe a medical practitioner combining both conventional risk factor with PRS. So I don't think that PRS is going to be like the only gold standard tool that is going to predict everything. Because, for example, like you mentioned, type 2 diabetes, for example, all those conventional risk factors are also key. So what I guess will be very relevant and useful is the combination of both conventional risk factor and PRS. So if I can make one quick comment about the communication of genetic to participants. For us in Africa, we are not at that stage yet where there is an exploration of PRS to participants, but we've done some genetic study in different parts of Africa, including from Uganda. And one major barrier is that it is always very, very difficult to even explain what's like the word DNA, for example, the word genetics or genomics. They are not even present in so many African languages. So it's very even difficult to even tell a research participant what you are doing. So in one study where we translated genetics or genomics to English, to the local language, and did a reverse translation. So you see the reverse translation was bringing out like a generational courses, which was totally different from the intention. You see, because there's no direct word for all those words. So in that particular settings in Africa, it's even more difficult to explain basic genetics to people, talk less of getting to the point of PRS.
Gil, did you want to give your perspective?
Yeah, just briefly, our experience of working, as Rahim was saying, with primary care practices in the northeast of the UK was that the information could be provided back both to primary care practitioners and through them to individuals who were already engaging in what is called health check, which is an assessment of overall health focused on cardiovascular health, which they typically have in midlife. Our experience was that through relatively light training and then the provision of substantial materials, both to the individuals and the GPs, that we could achieve very high levels of acceptance, satisfaction, comfort, and sort of benefit from all of those involved, both the healthcare practitioners and the individuals, without the need for clinical genetics support. And that's because in that part of practice, risk is already being used as a key tool within the management of individuals. And so by treating inherited risk, which most people are fairly familiar with, the idea that some of our risk we inherit from our parents, it becomes another risk factor amongst others. And that's it is actually the correct way of describing it. I'm all for not being a sort of genetics exceptionalist. I think it isn't the most important thing for many diseases. It is for some, and therefore it should be treated just as another thing that one needs to think about in the context of managing a patient. And our experience is, for many diseases, both in the UK and the US, that approach people are very comfortable with. Thanks.
Rahim?
I just wanted to answer the question in the chat a bit more. As I wrote about this, or I was asked about this bit in the article in the Times, so you can read that. But I did mention that the smartphone really is very important for our future health, both in terms of collecting data related to exposure, including diet. So the way we measure diet historically has been quite poor and with limited kind of validity. But using smartphones, taking pictures, using AI to analyze the food that we eat before and after kind of analyses looks to have great promise, actually, and would be a more accurate way of measuring diet. Similarly, for wearables, and measuring physical activity is much better than questionnaires, as well as more standard questionnaire kind of collection. But the smartphone will also be very important for feeding back that data and providing participants with a dashboard and engagement platform, etc., going forward. And finally, for interventions themselves. So for diseases, many diseases, as we know, non-pharmacological interventions, whether relating to diet, physical activity, tobacco, alcohol, etc., they're extremely important, particularly in early stages, when there may well not be a pharmacological intervention available anyway. And again, the phone can be used to provide those behavioral nudges or changes for something which is somewhat in its infancy. But again, there is some evidence, for example, the diabetes prevention program, that it can be delivered that way without requiring a kind of physical human being, either in person or online. So it's going to be extremely important for us going forward.
Thanks. So perhaps my last question, or my last thought, or for everyone to comment on, is that, we talked a little bit about how the statistical methodology might need to improve in terms of how we train polygenic scores, maybe to optimize what Gil was saying, basically trying to target whether we're avoiding harms, targeting benefits, or making equitable predictions across individuals, whether that might be across sex, strata, or ancestry continuums and things like that. But I wonder, do people think we need to just improve the representativeness of the genetic data to bridge the gaps in performance of risk prediction models? Or do you think there's something lacking in the current methods we're using that needs to be resolved first? Like, I guess currently we use polygenic scores that are based on additive assumptions where you just add up all the variants, but there might be other models out there that we should consider or use to sort of maybe bridge that gap further. I don't know if anyone had thoughts on that.
Yeah, I can start with this. I think that there is needs to improve representation. That is very key, particularly in African population where you have a lot of genetic diversity. So it's key that that population is represented. But to be able to get to that stage, that would take maybe like a decade of years to get to that stage where Africa is well represented. But why we do that? So one thought is that we know why PRS is not transferring to other populations. And one of the barriers has to do with the different allele frequency, different population. One of the barriers is the different effect size within population. I think what statistician can do now is probably to try to model those allele frequency of those individuals that are underrepresented. You know, the data is not available, but if you can model the idea allele frequency and the beta, maybe that could help at the moment. But that will not be like a standard of because if the PRS is being designed and with that in mind, and we neglect underrepresentation, we are not doing ourselves good. But at the moment, while we try to improve representation, I think statistical model can try to see if you can model those allele frequencies that are lacking, and as well as the effect size.
Gil?
So one of the key aspects of what we're trying to achieve here is that it's a prediction problem. So we're just trying to work out who's going to get the disease. And the great thing about prediction problems is that essentially the only thing that matters is how good it is at the end of the day. So in terms of models or the complexity of those models, or whether it's linear weights or whatever, I think one has to be empirical, and the best risk tools will be the ones that we should adopt. Now, what we mean by the best risk tools, I think, does need a bit of thought, because there are issues of performance, and there are issues of bias, and there are issues of equity in terms of discrimination, as various speakers have mentioned, and these are all important. So I think there's important questions about best, but given that once we define best, it's really just about the best model, where you need enough validation data, and I think that's the key thing here, enough validation data to be able to select the best. I mean, my own personal view is that we will end up with, rather than sort of PRS models, we'll end up with PRS or integrated risk kernels that draw in a whole bunch of sources of information, including genetics and other things, and have sort of, what's the right word, continuous evolution of the underlying parameters by context, the continuous context, and sort of more discrete influences as well, where those contexts are binary or categorical. So that's my own personal take, but I think the joy of it being a prediction problem is you just need the validation data, and then you'll know the answer.
Great. Yeah, that about brings us to time, unless or maybe I'll give Raheem one more chance to chime in if you want to add, because the last two they got their say.
I mean, just very briefly,
Closing
I'm
remarks.
not a statistician, so I can't comment on the models, but representative is certainly something I've thought about for decades in many ways. This even before we were looking at polygenic risk scores, there is a general problem of underrepresentativeness in clinical and medical research globally. And when for example, it was mentioned at the beginning of the study I set up in the UAE, at that point there was like less than 0.03% of the genomic data was from the Middle East. It's much better now because of various projects, but that's the kind of effort we need to make globally, to make sure that all neglected populations, the special efforts are made to include them, so that everybody can benefit from what are potentially transformational improvements to risk prediction and intervention and treatment. So we're making an effort to do that in the UK with our Future Health, but there needs to be a global effort to do that internationally as well.
Definitely. Yeah, we need to push on all angles to sort of make health and risk prediction more equitable for everyone, data and methods. Yeah. So with that I kept almost a perfect time, and with that I'll close this panel and hand it back to Bree.
Thank you, Sam, for chairing that excellent panel. Really, really insightful discussion from each of our panelists, touching on both the kind of system level factors of implementing polygenic scores in the clinic, and some of the more statistical things that we should consider as well. So yeah, thank you for everyone for attending. Let me just share my screen for just my last slide. Not going to take any more of your time today. I just want to highlight kind of what's next. So this is part of a a series of 2 workshops, the first one being last Monday on challenges to statistical approaches for health equity, which was kind of a precursor to this one, focusing on fairness in genomics and genomic medicine. I'm just going to pop a couple of links in the chat. It'd be really great to get your feedback on the workshop today. We are likely to run some similar workshops in the future, so it'd be great to hear, you know, what you thought was good, what you thought was bad, and how we can improve, and so on and so forth. These two workshops are part of a bigger piece of work looking at statistical methods for health equity and statistical methods for fairness in genomics. And if you want to stay up to date with this project, then well, you can sign up to the statistical methods theme mailing list, which is part of the data science for health equity community. And so there are two newsletters or mailing lists that you can sign up for there, and I've popped those in the chat. I've just realized I've sent those to Sam in a direct message and not to everyone here. So here are the links for both the survey and to sign up to the newsletters. And if you do want to add to those Jamboards that we shared in the links will be in the workshop survey form as well. So I'm going to leave it there. Thanks everyone for attending. Thanks to all the chairs for leading such excellent discussions, and for the panelists for their time and sharing their insights into all these important questions. And most of all, thank you to all the attendees for taking part and for putting such great questions to the audience. Yeah, please do stay in touch and sign up to our newsletters, and hopefully see you soon at another Data Science for Health Equity event. Take care, everyone.